James McCullough was in the lab extracting tiny cells called mitochondria from cells when researchers on his teams rushed in. They'd been operating on a pig heart and couldn't get it pumping normally again. McCullough studies heart damage prevention at Boston Children's Hospital and Harvard Medical School and was keenly interested in mitochondria. These power-producing organelles are particularly important for organs like the heart that have high energy needs. McCullough had been wondering whether transplanting healthy mitochondria into injured hearts might help restore their function. The pig's heart was graying rapidly, so McCullough decided to try it. He loaded a syringe with extracted mitochondria and injected them directly into the heart. Before his eyes, it began beating normally, returned to its rosy hue. Since that day almost 20 years ago, McCullough and other researchers have replicated that success in pigs and other animals. Human transplantations followed. In babies who suffer complications from heart surgery, sparking a new field of research using mitochondria transportation to treat damaged organs and disease. In the last 5 years, a widening array of scientists have begun exploring mitochondria transplantation for heart damage after cardiac arrest, brain damage following stroke, and damage to organs destined for transplantation. Mitochondria are best known for producing usable energy for cells, but they also send molecular signals that help keep the body in equilibrium and manage its immune and stress responses. Some types of cells may naturally donate healthy mitochondria to other cells in need, such as brain cells after a stroke, in a process called mitochondria transfer. So the idea that clinicians could boost this process by transplanting mitochondria to reinvigorate injured tissue made sense to some scientists. From studies in rabbit and heart cells, McCullough's group has reported that the plasma membranes of cells engulf the mitochondria and shuttle them inside, where they fuse with the cell's internal mitochondria. There, they seem to cause molecular changes that help recover heart function, when comparing blood and oxygen-deprived pig hearts treated with mitochondria to ones receiving placebos. McCullough's group saw differences in gene activity and proteins that indicated less cell death and less inflammation. About 10 years ago, Sitaram Imani, a cardiac surgeon at Boston Children's Hospital, reached out to McCullough about his work with animal hearts. Imani had seen how some babies with heart defects couldn't fully recover after heart surgery complications and wondered whether McCullough's mitochondria transplantation method could help them. During surgery to repair heart defects, surgeons use a drug to stop the heart so they can operate. But if the heart is deprived of blood and oxygen for too long, mitochondria starts to fail and cells start to die, in a condition called ischemia. When blood begins flowing again, instead of returning the heart to its normal state, it can damage and kill more cells, resulting in ischemia-reperfusion injury. Since McCullough's 8 years of studies in rabbits and pigs haven't revealed safety concerns with mitochondria transplantation, McCullough and Imani thought it would be worth trying in procedures in babies unlikely to gain heart function. Parents of 10 patients agreed to the experimental procedure, which was approved by the Institute's review board. In a pilot that ran from 2015 to 2018, McCullough extracted pencil eraser-sized muscle samples from the incision made for the heart surgery, used a filtration technique and isolated mitochondria, and checked that they were functional. Then the team injected the organelles into the baby's heart. 8 of those 10 babies regained enough heart function to come off life support, compared to just 4 out of 14 similar cases from 2002 to 2018 that were used for historical comparison. The treatment also shortened recovery time, which averaged 2 days in the mitochondrial transplant group compared with 9 days in the historical control group. 2 patients did not survive. In one case, the intervention came after the rest of the baby's organs began failing. In another, a lung issue developed 4 months later. The group has now performed this procedure on 17 babies. The transplant procedure remains experimental and is not yet practical for wider clinical use, but McCullough hopes that it can one day be used to treat kidney, lung, liver, and limb injuries from interrupted blood flow. It will take more research into the mechanisms of mitochondrial therapy, improved mitochondrial delivery techniques, larger trials, and a body of reported success before mitochondria transplants can be FDA approved and broadly used to treat ischemia-reperfusion injuries, researchers say. The ultimate goal will be to create a universal supply of stored mitochondria, a mitochondria bank of sorts that can be tapped for transplantations by a variety of healthcare providers. If you like more daily doses of positive news, please subscribe to my channel. Please also like and leave a comment, as this really helps the channel grow. Links to the article on this news story can be found in the description of this episode. Thanks for watching.